Well, now I'm chatting with Vince Lombardi, who's the scientific director of the Whittemore Peterson Institute, the WPI, and he's been in medical research for quite a while now in Philadelphia and some experience in Europe as well, if I'm not wrong, and so on. And he's had a very interesting paper out. This is he's done a lot more, but this year, this spring in March, I believe, we had a very interesting paper come out from the WPI with collaborators from around the world, amazing, Europe, Russia, so on, that I want to talk about because I don't know how to think about it. Where do we go next from there in terms of research? And how should I think about it now as a person who knows a little bit about science, but I don't really understand the context? And I'm leaving it to you to describe what the discovery was of the dendritic cells inside, so on.
Sure. Well, like any scientific discovery, it's a stepping stone to hopefully the next level. And so it's somewhat preliminary, but it gives us some direction because even though it was a small number of subjects, we found what we were looking at only in ME patients and none of the controls. So that was significant. Mathematically, it was significant. Basically, the genesis of this project was just some thinking about, well, what do we know about ME? And there's two things to me that are high on my radar. First of all,
Okay.
Right now there's a lot of talk about ME having similar overlapping symptoms with other diseases which are autoimmune in nature. Okay. The other issue is the fact that when I look at ME and those other diseases, what are some of the things they have in common? One of the things they have in common are gastrointestinal issues. When you look at a disease like MS or a disease like lupus, which you don't necessarily think of them as having gastroenterology or things like that, but they do. It's very common. So with that idea, myself and Dr. Demirlier conceived of this project that we were going to look at gut biopsies, and we were looking at...
Let me interrupt. I just wanted to say that Vince did this project with Dr. Kenny Demirlier from Brussels, who is very well known for his work over the past 25 or so years on myalgic encephalomyelitis and chronic fatigue syndrome. I just wanted to, excuse me, I wanted to highlight that so they got the point.
Sure. So anyway, one of the things that we were interested in looking at is that in all these other autoimmune diseases, for some reason, they find either expression of proteins or antibodies to endogenous retroviruses. So we went looking for that, and we found them in gut biopsies taken from the duodenum. And we identified them. We were thinking we might find them in all cells, but we ended up finding them in only a certain type of cell. And as it turns out, it was kind of like shooting fish in a barrel because it's a cell line I was very familiar with because I'd had an interest in it before because of its ability to produce interferon. So the cells we were looking at were a subset of dendritic cells called plasmacytoid dendritic cells. And they're the primary producers of type 1 interferon in the body.
Oh, now, if I'm not mistaken, we didn't even know there were, the world didn't even know there were dendritic cells until around 1996.
Well, they've known that the dendritic cells were around for a while, but this certain subtype of dendritic cell was only discovered recently. They've actually seen them in biopsies. Pathologists have described them as like a plasma type cell in biopsies from lymph nodes, but it was only recently, like 1996 I think it was characterized by Genscher's group that they'd identified previously a cell which produced a lot of interferon, but they had finally tied the two together. They realized these cells looked like plasma cells were in fact the professional interferon producing cells. And so the reason why I had an interest in the interferon-producing cells is because I had done work looking at cytokines in a large population of chronic fatigue syndrome patients, and although some of the chronic fatigue syndrome patients would spike high levels of interferon, statistically, over the big picture, they actually had less interferon than the healthy control people. And so that also got me looking at what other diseases have similar issues. And for instance, like MS, they treat MS with interferon, whereas lupus, they make too much interferon. So they're both autoimmune, but they have kind of opposing issues with plasmacytoid and dendritic cells, but at least got me interested in looking at those types of cells. So when we did these biopsies and our immunohistochemistry showed that we had endogenous retroviral expression in one type of cell, it became apparent what they were, and then we confirmed that obviously by doing some molecular biology.
And so these endogenous retroviruses, these are little pieces of viral DNA left over from history?
Basically, that's exactly right. If you look at our whole genome, probably half of our entire genome is just genetic junk. And most of this genetic junk is ancestral integrated retroviruses that have become in our germline. Some of them have different names, but there's a few of them that they're so close to being intact that they have specific names, like they call them HERVs, or human endogenous retroviruses. Viruses. So there's HERV-W or HERV-K, HERV-FRD. There's a whole different class of them. Some of these things have been implicated in pathology and other diseases. There was a paper published by Bridget Huber that suggested that one endogenous retrovirus could be activated by herpes virus and acted as a super antigen. So in fact, there is it's most commonly known in MS, for instance, that's associated with the expression of HERV-W. And in fact, there's even a company right now that's making a humanized monoclonal antibody against HERV-W as a treatment. So that suggests there is some possible pathology related to these things. I'm not convinced that's the case, but at least it was a good marker for saying that, hey, there's something different about these cells in the ME patients versus the controls. And so we've focused on that somewhat, and that's kind of what's leading our direction. Now, the reason why I think it's significant is that these cells are the only immune cell that have two professional roles. The first professional role is as interferon-producing cells, which is what they're most well known for, but they're also antigen-presenting cells, so they are dendritic cells. So, and the fact that they're connected to all these other autoimmune diseases, to me, in my mind, gives some benefit to the fact that there is some data support that chronic fatigue syndrome or ME subjects have some semblance of autoimmunity.
So they produce antigens, and what was their other professional duty?
They make type 1 interferon.
Type 1 interferon,
Yes,
okay.
these cells make 95% of all type 1 interferon in circulation.
And is that kind of interferon the kind that you give to Hep C patients?
Yes, it is.
Same
Yes,
kind,
exactly.
okay.
So, and notably, type 1 interferon activates NK cells. So if you didn't have enough, then that might explain some of the NK dysfunction. In fact, that's how they identify these cells, by their ability to upregulate NK function. So there's a number of kind of tertiary things that make me think this is relevant. Also, if you look at different cell populations and say, all right, what do these cells do? Where do you find them? What happens when they go wrong? What type of inflammation do they make? A lot of the things that this type of cell does is things we see in chronic fatigue syndrome.
Oh, such as the inflammation.
Well, yes, the inflammation. When we look at chronic fatigue patients, they make a certain profile of inflammatory cytokines, and these cells basically make everything we see. It's not saying that there's not other cells that can do that. There are, but the fact that when we did our work in the gut biopsies, that was the only cell that we could see that was different. So, and the fact that most of chronic fatigue or ME subjects have gastrointestinal problems. So, I think that a cell that is found in the gut of people, which is dysregulated, might potentially lead to pathology. So that's one of the things we're focusing on. The fact that it's an antigen-presenting cell, if there's something going wrong with it, then it makes sense why people might have some semblance of autoimmunity, because these are the cells that drive that. They're one of the cells that drive that. So.
Very interesting. So now, just on a practical level, where did these patients come from? Were they American or Belgian or?
Well, they're anonymous patients, so I don't know all, but most of them came from Belgium, yeah.
And they had to do endoscopies or something.
Yes, they did, yeah.
So you did that. You paid for that in the course of the experiment.
The subjects had endoscopies, or actually these had endoscopies done. The subjects, I won't say they're healthy control subjects. They weren't healthy. They were people who had gastritis, but they didn't have ME.
Yeah.
And so that actually, I think, validates the whole issue even more, because they're not the perfect prime health. You could say, well, maybe this is something that's common to other things if people have gastrointestinal distress, but these people had other issues, but they didn't express endogenous retroviruses in their PDCs. So
that was a good control group
absolutely,
too.
yeah. And most people who are healthy just don't go and get, a gut biopsy. So,
Right,
it kind of limits you to what you can get. But.
Well, and also most gut biopsies don't look for dendritic cells.
No. And it has been reported that this type of dendritic cell is important in other diseases in the gut. For instance, there's some papers that were recently published that in HIV/AIDS, for instance, it's known that people who have advanced AIDS have this decreased number of these cells in their periphery. And so the big question is, well, are they being killed by the disease, or are they redistributing to some other part of the body? And the prevalent hypothesis right now is they redistribute. So when they look at the animal model of HIV, which is the macaques, and they have SIV, which is simian immunodeficiency virus, which is similar to HIV, they have gastrointestinal problems, and they have a higher infiltration of these cells in the gut, so they redistribute.
Oh.
And so it is known that these cells can be found in reasonable numbers in the gut. In the ME patients, we found more in the gut than we did in the controls, but we also found they're expressing endogenous retroviruses, whereas the ones in the controls weren't.
Now, where does this lead you to want to go next? That you have found a statistically significant number.
Well, as it sits now, it's observational, but everything that goes through my mind is about mechanism. So we want to really know why they're different.
Yeah.
So we have some technology now that allows us to dissect out these cells and to look at individual cells and determine how these are different from either A, cells from people who don't have ME, or B, cells from the ME patients that aren't making the endogenous retroviruses. And so that's what we're doing right now.
So you're going to do the endoscopies on some, well, I guess you won't know in advance which ones aren't making it. So you're just going to, are you going to look back at the tissues of the ones you've already done? Or are you going to try to get a new group?
We can't really do so much of that because we had just a limited amount of tissue, because these are basically surplus clinical diagnostic specimens, and we had what we had. We knew they were ME patients, but we didn't know who they were, so we really can't go back to them and say, hey, can you give me some more tissue? However, because we found that in a reasonably number of subjects, it was like 60%, then that suggests that if we pick 20 people, then a little more than half of them are probably going to have the same phenomenon going on. We knew that all these ME patients did have GI issues, because that was the reason that motivated them to get the biopsy.
Right.
And so this may not necessarily pan out to all ME patients, but certainly ones who have their symptoms characterized by GI issues.
Now, do you have any sense, which you can then research, on what might have led to this configuration?
Absolutely, yeah. I can't really talk about it right now because I'm working on a manuscript that suggests that, but right now we do have a working model where we can turn on the endogenous retroviruses at will. And so we potentially have a cell model of this phenomenon, not necessarily a cell model of chronic fatigue syndrome, ME, but at least as a research tool that we can maybe understand if this is the same thing that's happening in the patients. And so that's right now the highest priority project I'm working on right now, is to understand if we can turn on these endogenous retroviruses, do we see the same things going on in the gut biopsies of the ME patients? And if it turns out that's true, then it gives us a whole wealth of information, because we can manipulate these things in the laboratory and say, all right, let's follow this, let's dissect out this biochemical pathway, and let's figure either out why is it being turned off or turned on, and how potentially can we address it.
So would this by any chance be the same line of research that Dr. DeMerle might have hinted to me might lead him to figure out how the factors affecting ME patients in the past turned into them having the disease?
It's very possible.
Might be related in some way?
I don't want to put words in Dr. DeMerle's mouth, because he's a clinician, and he has a lot more knowledge about what drives the disease than I do. However, I can tell you that it's possible that this might be caused by more than one thing, because clearly the expression of these endogenous retroviruses is associated with a host of autoimmune diseases, and some of them, for instance, like a perfect example is MS and lupus. In MS, they don't make enough interferon. In lupus, they make too much of it. Yet, they're both characterized by this dendritic cell dysfunction, as well as the expression of endogenous retroviruses. So although it's not going to be a nice clear-cut answer, and it may be completely different in ME, at least it gives us a focus point. So I think that one of the biggest areas of research in ME right now is finding biomarkers. Now, and that is obviously important because it validates the disease. But it's conceivable we may not find a biomarker anytime soon, but in my mind, I think to myself, well, there's enough things in the disease that we can focus on that are consistent, like NK cell dysfunction or like inflammation, that if we just focus on that and figure out why it's happening, what's the mechanism of this, then it'll lead us upstream to possibly even find a biomarker or figure out what's driving the disease, at least in some subsets of patients. And so, that's my philosophy. Is pick a point that is reproducible in the patient population and dissect out the mechanism, what's driving it, and that hopefully will lead us to the answer.
Yeah, kind of connect the path between the biomarkers.
Exactly. Follow the trail of breadcrumbs up to the source.
Right, or the yellow brick road.
Exactly.
But very interesting. I'm very interested because I have been having terrible gastrointestinal problems since my aforementioned surgeries that led to me being sicker. So I wonder if there's some connection. Now, another interesting point, there aren't many people doing this research who are looking at anything other than blood. We're not looking, scientists generally who are doing research on MECFS are not doing much with anything other than blood.
It's easy. I mean, blood is the easy sample to get. And take some blood. I can make some more.
Yeah.
It's not so invasive. I can probably draw blood on myself. It's not that hard.
Yeah.
Getting a GI biopsy is a little bit more traumatic, and so. But I think that we have to have an open mind and look at everything. I think obviously blood's important, and it's going to be. If we're going to look for diagnostics, it's probably that makes the most sense because it's an easy sample to get. But you follow the clues you have. Scientists are kind of like detectives.
Yeah.
And you don't look for, the clues under the street lamp that has the light because the lamp is burned out where the crime was committed. You got to follow the clues where they are. And ultimately, by following the issues in the gut and the dendritic cells, we might ultimately come to a place where we can actually start looking at the blood. But right now, I don't have enough data to follow conclusively just by looking in the blood. We have to look in other places.